So it's my great pleasure to start this session with a keynote speaker, and great honor to introduce my colleague and collaborator for many years, Louise. She's Professor Emeritus of University of California, Berkeley, member of National Academy of Sciences. And let me read some of the facts from her great career. So she got PhD in plant pathology from University of California, Davis. Did postdoc as physiological chemistry at University of Wisconsin, Madison. After which she started as assistant and an associate professor at the University of British Columbia. And then in 1999 joined University of California, Berkeley, where she also served as a chair of plant and microbial biology department for several years, right before she was a division director for environmental genomics and system biology division at Lawrence Berkeley National Lab. She has a long list of achievements, honors, awards. Here are some of them. Elected Fellow of American Society for Advancement of Science, Fellow of Mycological Society of America, and American Society for Microbiology. So she received multiple awards, including Alexander von Humboldt Research Award and the Robert Meisenberg Award for Fungal Genetics. She was a Miller Professor and Fran Dickinson Chair of Woods Science and Technology. And finally, as I mentioned, since 2021, she's a member of National Academy of Science. We've looked at different aspects of fungal biology, and over 10 years studied model filamentous fungi Neurospora crassa to understand mechanism of plant cell wall deconstruction and signaling. And more recently, she started looking at development of functional genomics, high throughput techniques for functional genomics for non-model organisms. And today's talk is along those lines. So we hope to hear great stories about high throughput functional genomics to investigate plant root-bacterial-fungal interactions. Please join me in welcoming Louise to the stage.
Thanks, Igor, for that kind introduction. So today I'm going to tell you about sort of my last hurrah before I actually leave the planet. And that is I've been the benefactor of Neurospora. And so there's an amazing community. We have a full genome deletion set. There's genetics and biochemistry available for this organism. But there are many, many fungal organisms, many of which I have an affinity for, where it's hard to do stuff. And so I wanted to develop methods where you could transfer the tools developed for more model organisms to non-model filamentous fungi. And so that's what I'm going to be talking about today. This isn't on, Wendy? Well, that's all right. Today I'm going to be telling you about an MCAFES project. So MCAFES is a scientific focus area funded by the DOE. It is led by Trent Northern and with help from Adam Dauschbauer. And the purpose of the MCAFES project is to try to understand, get mechanistic understanding of fungal, bacterial, plant-root interactions, and that's the goal of the SFA. Great. So the organism I'm going to talk to you about today is trichoderma. And so trichoderma is a rhizosphere fungus, and it has some really interesting biology. It colonizes roots. It's an endophytic fungus. That is, it means it grows between plant cells. And when it does that, it actually primes the plant so that you have increased disease resistance, and it also enhances plant growth. And in fact, trichoderma is a commercially very important organism because it's added to seeds for enabling better plant growth. And so there is a lot of commercial interest in the genus Trichoderma for plant growth promoting properties. Now, trichoderma, as I said, it grows as an endophytic fungus, but in the soil, and probably also in the plant, it secretes a bunch of secondary metabolites, just like many other filamentous fungi. And the role and function and structure of these secondary metabolites in filamentous fungi is mostly unknown. It also secretes volatiles, and the secretion of the volatiles and the secondary metabolites affect root, but also other microbes in the environment. The other thing about trichoderma is that it's also a mycoparasite, and so it's helping plant growth and it's killing other fungi in the soil. And so again, this is a really interesting aspect of the biology of these organisms. Here you can see trichoderma coiling around the hyphae of a plant pathogen called Rhizoctonia solani, and it breaches its cell wall and kills the fungus and then secretes all the nutrients from the fungus. And so we were very interested in trying, again, to try to understand mechanistic interactions of trichoderma with other organisms in the environment, other fungi, and plant roots. Okay, all right. So as I said, filamentous fungi like trichoderma, there are 43 predicted biosynthetic gene clusters that form secondary metabolites in trichoderma. Most of those have not been characterized on a structural level, so we don't really know what they encode. And so we wondered whether, we wondered whether if we added exudates of trichoderma, whether it would affect the growth of rhizosphere bacteria. And as you can see, as you increase the concentration of the exudates, the growth of the bacteria is really lost in these cultures. And so we had access to these wonderful RBT and seq, random barcoded T and seq libraries developed by Adam Dauchbauer and Adam Arkin. And these libraries are in rhizosphere bacteria, and so that was really a focus of Adam's work. And what we decided to do is use these RBT and seq libraries. And so these are insertional libraries in transposons, and each insertion is uniquely barcoded. So then you have a library of mutants, and you can do fitness experiments, and instead of seeing what mutants go up or down, you actually see what barcodes that mark that insertion go up and down under particular experiments. And then you can assess, compared to the control, what genes are important for a particular function. And so we use these libraries using control media and spent media, and mapped, and then just did these bar-seq experiments. And the advantage of the bar-seq experiments is that you have these universal primers that span the unique barcode that is linked to the insertion. And so it's a very cheap, $10 cheap PCR reaction, which you then sequence. And so you can do a zillion, once you have the insertional library, you can do a lot of bar-seq experiments inexpensively. And so these are just some of the results. And so what we determined is that based on mutants that have lost fitness, which are on this axis, that trichoderma and bacteria are fighting for iron. And since these are exudates, this suggested that trichoderma secretes a siderophore, which was heretofore unknown, and that siderophore is sequestering iron, and that these mutants in the TUN-B transport system were unable to access that siderophore bound to iron in the media and bring it in and survive. And so these data suggested that trichoderma in the environment was, and bacteria fight for iron. And that's well known in many systems. So that wasn't terribly surprising, but it was also confirmation that this system is probably working. In addition, we also identified the fact that bacteria also are mutants in resistance to antibiotics were also dropped out in these fitness experiments when you compared these bacteria grown in regular media compared to media with the spent media from trichoderma. And these are involved in modifying both the plasma membrane, the membrane system, and the LPS to prevent these cationic antimicrobial peptides from entering into the cell. And so again, these data suggested that with that spent media, that trichoderma was secreting antibiotics that were affecting the growth of the bacteria. And many of these are involved in protection against an antibiotic called polymyxin. Polymyxin had never been identified previously in Trichoderma atraviridae. And when we added polymyxin to our libraries, we ended up getting the same genes that were identified in the spent media of Trichoderma. So these data indicated that Trichoderma was making a polymyxin or a polymyxin-like compound out of those 43 biosynthetic gene clusters. And so again, we not only learned something about the bacteria, but we also learned something about fungi, about the compounds they make in these cultures. We also decided to go in and look at individual mutants to show that our data that we identified from fitness experiments was actually real. And so fortunately, the JGI had arrayed all of these insertion mutants, and we went in and looked at insertion mutants for some of the ARN pathway, which is resistance to polymyxin, and the MPR pathway, which changes to phospholipids. And all of those mutants were very sensitive in both Pseudomonas simiae and Pseudomonas putida, very sensitive to those to the spent media of Trichoderma. And so that indicated that the fitness data that we identified, where we're just looking at these random barcodes going up and down, when you look at individual mutants, you get the same result. Again, recapitulating the data from the fitness experiments, which was really great. Okay, as I said, Trichoderma has 43 biosynthetic gene clusters shown here, and Manuel, who was the author of this paper and did all the RBT and seq studies in the bacteria, also using 300 RNA-seq experiments, characterized the expression of these biosynthetic gene clusters. And it was known, or he identified the fact that mutations in this TMK3 pathway, which is the HOG1 pathway identified in Saccharomyces, actually affected expression of many of these secondary metabolism clusters, so they were lower. So we predicted that spent media from this mutant would not have the same effect as spent media from the wild type. And so that's exactly what we found. You see here on the left, I guess it would be your left here, that again, you have all of these mutants in the pathway associated with resistance to polymyxin and transport, and all of those are gone in the spent media from this TMK3 mutant. And so these data indicated that the TMK3 mutant was very important for expression of some of these secondary metabolism clusters, probably not directly, but indirectly, that affected the response of the bacteria to these compounds. And so we did mass spec, and you can see here on the left is the blue, and that's the profile, the mass charge of the mutant, and it makes much smaller compounds compared to the wild type, again showing that in this mutant you have a huge change in metabolism, and particularly secondary metabolism, because these were the exudates that we identified from Trichoderma, wild type and mutant. Okay, so as I said, Trichoderma makes exudates, secondary metabolites that are present in exudates, but it also makes volatiles. And this is very common in filamentous fungi. And in fact, when you open up a plate of Trichoderma and smell the spores, it smells like coconut. And that's because of this 6-pp, which is well characterized in Trichoderma. And so these are volatiles. Kat Adams, who a postdoc in my lab, characterized the volatiles based on the literature which was kind of a mess, from Trichoderma. And we wondered whether these volatiles would also affect bacterial growth and interactions. And so she developed what she calls the fungal thunderdomes. And so here we have an empty plate, and the bacteria growing, they are not touching physically, so they can only interact via these volatiles. And here is a plate inoculated with Trichoderma, and you can see it really severely affects the bacterial growth in this case. And this is just a graph showing that when you use these thunderdomes, it reduces bacterial growth, not of some bacteria quite significantly, others less so. So the thing about these volatiles is that in the thunderdomes you have a mixture of volatiles that are being released, very much like the exudates. And so many of these compounds that Kat identified in this tree here were commercially available. And so we decided, rather than looking kind of like a mixture in the response of bacteria, we decided to use individual compounds. And so you can see here that bacteria respond very negatively to a lot of these volatiles from trichoderma. Some are very sensitive, like azospirillum, to everything, others less so, like Klebsiella, for example. And so we decided to do the RBT and seq libraries with bacteria using volatiles from trichoderma. And we really wanted to have about 50% inhibition of growth. And that's because, this is a caveat with these RBT and seq libraries, you need a pretty strong response to get something, to get statistically supported data. And so Kat did that. And so these are the 5 libraries where we have RBT and seq data from Bosea, Rhizobium, Lysobacter, Azospirillum, and Xenorhizobium. And so you can see that in some cases you have an overlap of the responsive genes to a bunch of different compounds, but then others are unique. And so we're just going through this data. Kat just got this data for these 4 species recently. And so we're interested in both responses that are unique to a particular bacteria and also responses that are across all the bacteria. And so again, we wanted to do a proof of principle to see whether these RBT and seq data recapitulated the volatile results. And so Kat and Mitch Thompson made a mutant in one of the Bosea targets that we identified by these fitness libraries. And you can see here that the mutant, it's in a TET-R gene, so transcriptional regulator, and it's very sensitive now to these compounds compared to wild type. And what's important is that it's also sensitive in the fungal thunderdome assay. And so when you take, these are the fungal thunderdomes, so Trichoderma atraverde, and you can see you get a reduction in wild type, but when you have this mutant, you have a much greater reduction. Again, recapitulating our fitness results that these volatiles are affecting certain pathways in the bacteria that work as individual compounds and then also in the volatiles from trichoderma. So this is pretty exciting because for me especially, because very little is known about the interaction of volatiles and other organisms in the environment, and so this provides a way to get at that interaction. So just to summarize this part of the talk, trichoderma exudates contain an environment where microbes fight over iron. Species in the genus Pseudomonas have antimicrobial resistance proteins that are very important for interaction with exudates from probably a number of filamentous fungi. And the use of these RBT and seq libraries have been really a gift to understand how fungi and exudates and volatiles shape rhizosphere bacteria. Okay, so that's all great, but I'm really a fungal biologist. I've been learning, I actually did my PhD on bacteria, but I really wanted to be able to ask these same questions in a fungus. So that's a challenge. There have been random barcode tDNA-seq libraries in uninucleate yeasts like Rhodosporidium and Saccharomyces, but not for filamentous fungi. Filamentous fungi are multinucleate, syncytia. They grow as filaments. It's much more of a technical challenge. So today I'm going to tell you about our progress in this area. The first thing we did is we generated a plasmid that has, again, it was based on the RBT and seq. You had these universal primers that can amplify unique barcode and a resistance marker. And so we use hygromycin and erythromycin in this case. So we have 2 plasmid libraries. That's transformed into your favorite fungus. So this is trichoderma on one side, neurospora on the other. And then you map where those barcodes are, just like you do with the RBT and seq data. So you have a unique barcode associated with an insertion. And then you can do, once you have this library, you can take that library of mutants and do bar-seq experiments, just like in bacteria, to identify what barcodes go up or down depending on the experiments that you're doing. And so one of the important things is that, too, is increased transformation efficiencies, because the genomes of filamentous fungi are much bigger than bacteria. And so we wanted about 10 insertions per gene, which is about 1 million transformants. Well, we haven't really quite gotten there. This is like what we hope to get eventually. But we were able to increase transformation efficiency so that we can get 60,000 transformants in about a week and a half, which is huge. And 60,000 transformants, considering there's about 10,000 genes in these genomes, you'll see you get hits in almost every gene in the genome. And so that effort, led by Lori Huberman and Manuel, we now know what to tweak in terms of your favorite filamentous fungus to increase transformation efficiency. So that was huge. The second is a little bit about this library. Again, we had 8 million unique barcodes in the E. coli library. When you transform it into agrobacterium, you lose some, but we have 140 million plasmids, each that have a unique barcode in them, and that's used for transformation of filamentous fungi. This is a library that was constructed by Lori Huberman, and we hope to make this library available to anybody who works on filamentous fungi along with their agrobacterium transformation technique to make your own personal library. So the mapping part is kind of expensive, but once you have everything mapped, you never have to remap it again, and then you can use that fitness library for all of your bar-seq experiments. And so again, we decided to use Trichoderma atraverde. This is a genome hosted by the JGI and updated a little bit by Alfredo Herrera Estrella. There's around 11,000 genes. There's a lot of long non-coding RNAs, which are interesting, and microRNAs. And most of the genome is annotated based on other organisms. So these 2,500 with biochemical pathways are really kind of like Saccharomyces. And there's only a few mutants that have really been characterized. And they're in conserved pathways where their function is similar to what has been characterized in other filamentous fungi. And so again, a lot of unknown in this. And so we had about 60,000 transformants. We mapped all those insertions. And we have unique barcodes in about 7,100 genes. Now what's important for this, there's two things that are important. First of all, you won't recover essential genes in this because the conidia are homokaryotic and uninucleate. So if it's an essential gene, you won't grow. And so presumably in that 7,100 to the 11,000 predicted in the genome are the set of essential genes for Trichoderma and filamentous fungi in general. Secondly, it's annotation independent. You get insertions across the genome. We have insertions from telomere to telomere. And so if you have an insertion in a desert where there's no annotation and it has the insertion there, that barcode goes up or down depending on the environment or a test that you've done, then that is telling you something about that region which you don't know about. And so the great thing about this is that it's not, the great thing about these bar-seq experiments with filamentous fungi is that it's annotation independent. So we think we'll get a lot of information just about genome function by these experiments. Let's see. And so these are the insertions. They're primarily intergenic, but we definitely get a good insertion within annotated genes. And they span all of the categories associated in the genome annotation, the GO categories. All right, so we had things inserted, 7,000 genes mutated. Can we do bar-seq experiments? And again, this is challenging because it's filamentous, it's not uninucleate, it's not a single yeast cell. So it actually took us a long time to figure out what conditions to do these under. But we have a test condition and then your experimental conditions. And again, we map the insertions and then we do bar-seq and we look for what barcodes go up or down depending on the condition. And so we decided to use carbon. And so that was a great suggestion by Adam Dauchbauer because that works really well in bacteria. And we just wanted a proof of principle. Can we do bar-seq experiments and uncover mutants using this experiment? And so we did carbon sources and amino acid sources. And so what we have here are Trichoderma atraviridae grown on different carbon sources. And you can see here the replicates, the triplicates actually, are very similar to each other, which was really comforting because in these conditions, you can imagine even a slight skew would really change your data. And so after much work, we've identified methods where we can get reproducible data from triplicates, biological triplicates. And so again, we used carbon sources and amino acids. And so we found that insertions in genes known for the utilization of xylose totally dropped out. And so again, this was a test. And so these are the barcodes. And you can see for xylin R, which is the regulator here, those mutants totally drop out. And then some of the enzymes associated with xylose utilization, xylitol dehydrogenase and xylokinase, those mutants also drop out on xylose, but we're pretty good in the other conditions. And so this is a map, and so in here are a bunch of HP, which are hypothetical proteins, and then there's glycosyl family 15 here at the top, and so we made a mutant of that particular gene in Trichoderma, and it had reduced fitness on xylose. So even in things that are pretty well characterized, we identified new components that were important for utilization of xylose. And we leveraged the Neurospora data for this. I mean, this is a conserved pathway in filamentous fungi. And so we looked at the mutants of the orthologs of these genes in Trichoderma and Neurospora and went to our library, pulled out the mutants, got this data in a day, none of them grew. And so again, recapitulating the fitness data. And these are Barsic experiments with cysteine biosynthesis, and so we always add dropout mix to all of our control plates. And so this is dropout mix on the top, on the top, and dropout mix missing cysteine on the bottom. And as you can see here, all of these genes associated with cysteine biosynthesis had less fitness in media dropout mix with all the amino acids except cysteine. And so again, we uncovered mutants in genes that were very important for cysteine biosynthesis. So again, recapitulating that this system really works. And so what we're excited about doing is extending this to more unknown territory. So interactions with bacteria, interactions with plant roots to try to identify novel functions associated with and also host-parasite interactions between Trichoderma and its prey, fungi, to try to identify new components of function of Trichoderma. Okay, so our ultimate goals are to be able to use this random barcode DNA-seq approach to assess the role of genes and pathways in Trichoderma that are important to the interactions with bacteria and plant roots, and we really hope to extend this, these insertional mutant libraries to filamentous fungi in general, and we think that will really be an amazing way to interrogate fungal whatever interactions. And then we're hoping to develop high throughput ways to confirm the fitness of our mutants by insertional libraries. Am I doing okay time-wise? Oh, okay. To enable high throughput functional genomics of, I think I'm a little bit far, but anyway. So these are the people that I want to acknowledge that have really contributed to the work. Manuel and Lori. Lori did all of the library construction the construction of the plasmid and the understanding of what's important for agrobacterium transformation, which took a year, going through all the different components and figuring out what to do to increase transformation efficiencies. Manuel did all of the RBT and seq with bacterial libraries and also constructed the Trichoderma mutant library. Kat is doing all of the volatile work. Vayu, who just got a job at Stanford as assistant professor, is continuing to work on MCAFEs, developing methods for very high throughput functional mutational analysis in filamentous fungi, again, that can be extended to many other species. And I just want to acknowledge Trent, Adam Dashbauer and his fabulous libraries, which have just been such a pleasure to work with. Everything is run by Astrid. And collaborative work we have with Kate Zelnina, where we're taking some of these mutants, individual mutants, and looking at them in terms of their function compared to wild-type trichoderma mutants and mutants in trichoderma, like TMK3, to see how that shapes bacterial interactions in plants in ecopods, which are structures or incubation that recapitulate field conditions. And again, our organism, trichoderma. And also we're sending all our data to Daniel Segre. He's a modeler to try to understand how these affect the metabolism of filamentous fungi and help develop models for us to understand that better. And with that, I am finished. I'd be happy to take any questions.
Thank you very much, Louise. We have some time for questions. Let's start from that side.
Hi, really interesting talk, thank you. I was curious about the polymyxin story. So it would be really exciting, you know, if the fungi is producing a polymyxin typically produced by Pinnibacillus species. I was wondering if of those 43 BGCs, if you've actually found that gene cluster in trichoderma, in RPS substrate prediction, you know, hallmark of polymyxin's incorporation of diaminobutyric acid. Just as a natural product nerd, that would be really fun. So I was just wondering if that's been validated
that's
at all.
a really great question. And so one of the things we're doing is, you know, as I said, developing these high throughput mutational analyses. So the thing about the fitness is that anything that's secreted, mutants in that particular product won't come out in the fitness. And so we're going in and we're mutating each individual 43 biosynthetic gene cluster in trichoderma. And then we can use the bacteria as a readout, right? So which mutations in which of those clusters? There's one that kind of looks like one, but we can use the bacteria as a readout. Okay, so you have this single mutant. When you use exudates from that single mutant, how does that affect the readout of genes that we identify in the bacteria? And in the bacteria, you know, I showed you things that we knew what they were, but there's a bunch of hypothetical. So a huge amount of data that can be mined for a long time to figure out, you know, what's going on there. So that's actually Vayu's project at Stanford, is to make these high throughput, and we've focused on the biosynthetic gene clusters as a primary. We're also collaborating with a group in Denmark who are secondary metabolism structural people, and so they've been trying to identify the actual structure of some of these. But I think those are really complementary approaches. Great question, thank you.
Some questions in the middle of the room. Yes.
Hi, Louise. So looks like you mentioned the trichodermal things, like you said the DNA library covered around like 7,000 genes. I'm wondering if it's possible to develop some approach to cover the rest of the genes in the genome.
Yes, absolutely. So we decided to do, you can consider this really almost as a test library. Because once we had the library, then we had to work out the conditions for the bar-seq and et cetera. And so if there were going to be major complications with doing bar-seq experiments with filamentous fungi, we wanted to know earlier rather than later. And so what I've shown you is a test library, but Manuel is making, getting better coverage across the genome so that we should have everything at saturation, so that will tell us the essential genes because we won't find any insertions in those genes, but we'll have saturation across the genome. And so what we really want is that, you know, maybe 5 to 10 insertions per gene, and those barcodes should go up and down together based on the treatment that you have. And that just gives a lot more statistical power to your results than having, for example, a single insertion in a gene. So those experiments are ongoing right now. Actually, I think some of them are in the sequencing queue.
Thank you.
Mm-hmm.
Hi, thanks. That was a fantastic talk. I'm interested in the VOC portion, and it seemed like with that story that most of the bacteria responded negatively to the VOCs, and that kind of surprised me. I wonder if that surprised you too, or if that was the experimental design. I'm just wondering if there's some bacteria that might specialize in eating the VOCs. Maybe there was enough organic carbon provided so that you wouldn't have seen those, but were there any that you saw that responded
Yeah, that's
positively?
a great question, and Kat actually addressed that and tried to make it where they couldn't do anything but eat that particular VOC. She never really got anything that was, that we could really say yes. I think that's true, and I think that just like any signaling compound, it could have differences in the bacteria, and that makes their response different, and so you could have bacterial interactions depending on the response of your VOCs. But we did try to see whether anybody ate anything, and we were not able to really show that. Some of them had a little bit of a positive response to the Thunderdomes, but when we added the individual compounds, that wasn't recapitulated.
Thank you.
Great question.
Thanks, Louise. A lot of great, fascinating stories. I was curious about those non-coding RNAs. Did you see any TNC hits to those?
Yes, absolutely, and also microRNAs.
I see.
Absolutely.
Okay, so those are in your mix, and I
Yeah.
guess that you probably have some data that's interesting for all your different biology there, right? So
Sorry?
you probably have results there that are interesting for VOCs and all the other biology, right? So they're just
Yeah,
like another gene.
Yeah, VOCs are a little bit trickier, just like the exudates are trickier. If you have mutations in those genes for the tDNA-seq, for the fungal part, I'm not sure this is your question, you won't see those in your results because anything that is secreted or volatilized, you'll get complementation. And so that's why we're making individual biosynthetic gene cluster mutants, because those would not be recovered in the fitness experiments. And if we knew more about the pathways and if the genes weren't essential for those VOCs, or if we could alter them experimentally, then we could start to address those that as well. So in other words, have a mutant that really affects your VOC profile, and see how that affects bacterial interactions, for example. But the VOCs are harder to think about how you'd address their biosynthetic pathway mutants, unlike the BCGs.
Thank you.
But it's unknown. It's pretty cool.
Any other questions?
Beautiful talk. How about fungi sensing these bacterial products? How fungi get the information that there is something out there? Are they bacterial cells? Are they sensing these compounds? You mentioned the Hog pathway. Could you elaborate on this a little bit?
Yeah, that's a great question. I mean, right now with the test library, we just wanted to make sure we worked out methods, which took a long time to get very consistent, robust results. But we might do bacterial exudates against the tDNA in the trichoderma library to answer that very question. What is it that bacteria secrete that affect the response of trichoderma? And so, yeah, that's definitely on the table to kind of look at those interactions. And so many people, as I said, people have looked at trichoderma in plants and a little bit about trichoderma as mycoparasites, but if you think out in the rhizosphere, the organisms that it's going to interact with the most are other bacteria. And very little is known actually about how trichoderma shapes bacterial communities, how those bacterial communities affect fungal interactions, and so we hope to kind of get at mechanistic components of those interactions via the MCAFEs project.
Thank you.
Anybody else? I have a final question, maybe, or two. So, ideal situation to understand function of the gene is deletion of single gene. How frequent is this insertional mutagenesis affect multiple genes at a time? So, and what's the solution for that?
Well, if you have multiple insertions in a genome, it's very hard to tease apart. So I actually talked to Charlie Boone about this, and I was telling him that we just throw those away. But Charlie Boone is a yeast biologist, and he looks at synthetic interactions of mutants in the Saccharomyces, and so he's made all of these synthetic lethal arrays. And so if you have one mutation, it's fine. You have another mutation, it's fine. But you put those mutations in the same genome, it's wiped out. And so he was saying, don't throw those away, because if you have a mutation elsewhere in the genome and you get a different result because you're having a readout of 2 pathways, it might actually tell you something about genetic interactions. I think we have to have a much higher frequency of insertions across the genome to be able to tease that apart. But I had never really thought about that. And so that's something interesting to think about, that you could actually look at interactions in the pathways if you had a particular transformant that had more than one insertion.
And sort of to continue on this line, so most of the pathways are redundant, right,
Right.
in fungi. So once you, what's the best solution there? So can you take a mutant and apply the same strategy and have double, triple mutants?
Potentially. You mean take a mutant with a single insertion and then
That is
re-mutagenize it? Yeah, yeah, definitely. Again, I think of these, it's at a point where it's not hard to do. It's one of those projects that's technically challenging and boring.
Right,
You know?
okay.
Because it's very easy to get bottlenecks, you know, and so you have a bottleneck when you do the transformation and you get tons of insertions, but it's the same barcode. Well, that doesn't help you at all. So anyway, but I think these, you know, to be able to do those methods, you can start to do things like that, and they are definitely not outside of the realm of
Okay, great.
possibility.
Thank you very much. Let's thank Louise again. Wonderful work.